Good morning, everyone, and welcome. I am pleased to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in pharmaceutical sciences. Today, we will be discussing the process validation of active pharmaceutical ingredients, or APIs. Over the course of this presentation, we will explore the key aspects of validating the processes involved in API manufacturing, ensuring quality and compliance. So, let's get started and dive into this important topic together. Let's start with the definition. When we talk about process validation for APIs, we're referring to the demonstrated evidence that a process consistently produces an API, meeting its predetermined specifications and quality attributes. In simple terms, it's about proving that our process reliably delivers a product that meets the set standards every time. Regulatory expectations put a strong emphasis on several key areas. First, risk assessment is crucial, understanding and managing potential risks throughout the process. Then, there's the control strategy, which is about having a well-defined plan to maintain process control. And of course, making data-driven decisions ensures we base our actions on solid evidence rather than assumptions. Now, looking at the regulatory references, we see important guidelines like ICH Q7, which covers GMP for APIs, and the FDA's process validation guidance, which adopts a life cycle approach. This life cycle approach highlights that validation isn't a one-time event, but an ongoing process throughout the product's life. In summary, process validation is critical to ensure quality and compliance, and it's supported by these key regulatory frameworks that guide how we establish, monitor, and maintain process control. Now, let's walk through the typical flow in API manufacturing. It all starts with dispensing, where the raw materials are measured and prepared accurately. From there, we move into the reaction or transformation stage, where chemical changes occur to synthesize the active pharmaceutical ingredient. Once the reaction is complete, the next step is isolation, where we separate the API from other substances. Following that, purification helps us remove impurities, ensuring the API meets quality standards. Then, the material is dried to achieve the desired moisture content. After drying, milling reduces the particle size, which is crucial for consistent formulation and bioavailability. Finally, the API proceeds to packaging, where it is prepared for distribution. Each step in this flow is vital to ensure we produce high-quality, effective pharmaceutical ingredients, aligning with our overall goal of delivering safe and reliable medicines. Now, let's take a closer look at dispensing and material handling, which is a critical part of our scientific focus. The main goal here is to ensure the raw materials are correctly identified and accurately measured. This helps us prevent any mix-ups or errors that could impact the final product. Another key point is preventing cross-contamination. We have strict procedures in place to make sure that different materials don't accidentally get mixed or contaminated, maintaining the integrity of our processes. Traceability is also essential. We keep detailed records of weighment and witness checks so that every step is documented and can be reviewed if needed. This creates a transparent chain of custody for our materials. For example, our standard operating procedures for dispensing include verifying the raw material labels twice, by two different operators, to double-check accuracy. We also use calibrated balances, typically with an accuracy of plus or minus 0.01 grams for quantities under 1 kilogram, to ensure precise measurements. Control tools like checklists help us stay consistent, and we set weight variance tolerances, usually around plus or minus 0.5%, to catch any deviations early. All of these practices work together to support our overall goal of quality, safety, and traceability in our manufacturing process. Let's talk about risk assessment. The main purpose here is to help us prioritize which critical processes or attributes we should focus on, especially based on how much they impact quality. By doing this, we can ensure we're tackling the most significant risks first and making the best use of our resources. Now, to do this effectively, we have a couple of powerful tools at our disposal. One is FMEA, which stands for Failure Mode and Effects Analysis. This helps us identify potential failure points in processes, evaluate their severity, and determine how likely they are to occur. It's a proactive way to spot risks before they become problems. The other tool is Ishikawa or cause, effect diagrams. These are great for digging into the root causes of issues. By mapping out all possible causes, we can better understand where problems originate and develop targeted solutions. To put this into perspective, imagine we're working on a manufacturing process. Using FMEA, we might identify a potential failure in a component that could affect product quality. Then, with the Ishikawa diagram, we could explore all possible causes for that failure, such as equipment issues, human error, or material defects. This comprehensive approach ensures we're addressing risks systematically and thoroughly. Overall, risk assessment is about focusing our efforts where they matter most, and these tools help us do just that, making our quality management more effective and proactive. Let's take a look at critical process parameters, or CPPs, and critical quality attributes, known as CQAs. First, CPPs are the process variables that can influence the CQAs. In other words, these are the factors we can control during manufacturing that directly impact the quality of the final product. On the other hand, CQAs are the attributes that we need to monitor and control to ensure the API meets quality standards. These attributes are essential because they define the quality profile of the active pharmaceutical ingredient. To put this into perspective, imagine a manufacturing process as a recipe. The CPPs are like the ingredients and cooking conditions you can adjust: temperature, time, mixing speed, while the CQAs are the qualities you want to achieve, such as potency, purity, or stability. Understanding the relationship between CPPs and CQAs is crucial because it helps us identify what we need to control tightly to ensure consistent product quality. Now, let's look at an example to see how this works in practice. Let's talk about in-process checks, or IPCs, and why they are so important. Essentially, IPCs help us make sure that the process stays in control throughout production. By doing so, we can reduce the chances of out-of-specification events, or OOS, which could compromise the quality of the final product. Now, some common types of IPCs include measuring pH, monitoring temperature profiles, checking the reaction endpoint, often using techniques like TLC or HPLC, and performing weight checks after isolation. These checks are critical because they give us real-time information about how the process is progressing and whether everything is within the desired parameters. Let's look at an example, such as pH control. Suppose our target pH is 7.2, with an acceptable range of plus or minus 0.2. We measure the pH every 30 minutes to ensure it remains within that range. If it drifts out of range, we can take corrective action immediately, preventing larger issues down the line. Overall, these checks are vital for maintaining process consistency, ensuring product quality, and ultimately supporting compliance with regulatory standards. They keep us proactive rather than reactive, and that's key in pharmaceutical manufacturing. Let's talk about sampling strategy, focusing on both in-process and final sampling. Regulatory expectations emphasize the importance of representative sampling, meaning the samples we collect should accurately reflect the entire batch or process. This helps ensure the data we gather is meaningful and supports quality assurance. It's also crucial to have a documented rationale for our sample size and sampling frequency. This documentation shows that our sampling plan is well thought out and justified based on risk, process variability, and regulatory requirements. Now, when it comes to a typical sampling plan, the key points are to determine how many samples we need and how often we should take them. These decisions should be based on scientific and regulatory considerations, ensuring our sampling is both effective and efficient. Remember, a solid sampling strategy helps us maintain product quality and compliance. It's about being systematic, transparent, and aligned with regulatory expectations, which ultimately supports the integrity of our manufacturing process. Now, let's look at how we validate our sampling plan with a practical example. Imagine we have a batch size of 1,000 kilograms, and we're sampling from 10 equal lots. Based on regulatory expectations, which are risk-based, we've decided to select 10 samples for the final API. The key here is that our sample size isn't arbitrary. It's guided by the need to ensure product quality while maintaining efficiency. When it comes to making acceptance decisions, the rule is straightforward. If 8 out of the 10 samples meet the criteria, we can consider the batch acceptable. However, if there are 3 or more out-of-specification results, or OOS, that triggers an investigation. This approach helps us balance quality assurance with practical sampling, ensuring that we catch potential issues without oversampling. Remember, this validation process is essential for maintaining confidence in our quality control system and aligning with regulatory standards. It also supports consistent decision-making across batches, which is critical for our overall production integrity. Let's take a closer look at statistical process control, or SPC, which is essential for monitoring and controlling manufacturing processes. On this slide, we're focusing on some common metrics used in SPC, specifically CP and CPK, which measure process capability, and control charts, which help us visualize process stability. Starting with CP and CPK, these are key indicators of how well a process can meet specifications. CP, or process capability, compares the spread of your process to the allowable specification limits. Essentially, it tells you if your process has the potential to fit within those limits. CPK, on the other hand, adjusts this measure based on how centered the process is within those limits, giving a more realistic picture of actual performance. Let's walk through an example calculation to illustrate these concepts. Imagine we have API assay values from validation batches, with a mean of 100% and a standard deviation, or sigma, of 1%. The lower specification limit, LSL, is 97%, and the upper specification limit, USL, is 103%. To calculate CP, we take the difference between USL and LSL, which is 103 minus 97, and divide that by 6 times sigma. So that's 103 minus 97 divided by 6 times 1, which equals 1. This indicates that the process has the potential to be capable, but depends on how centered it is. Now, for CPK, we look at how close the mean is to each limit. We take the minimum of 2 values. One is the difference between USL and the mean, divided by 3 sigma. The other is the difference between the mean and LSL, divided by 3 sigma. Since both calculations come out to 1 in this case, the process is well-centered and capable. Overall, these metrics help us assess process performance and identify areas for improvement. They're fundamental tools in SPC and tie back to our goal of ensuring quality and consistency in manufacturing. Now, let's take a look at the final stage of our analytical testing process. This slide covers the required tests we perform to ensure the quality and safety of our active pharmaceutical ingredient, or API. First, we have the assay, which is typically done using HPLC. This helps us determine the potency of the API. Then, there's the impurity profile, where we identify any impurities present in the sample. Residual solvents are checked using gas chromatography, or GC, to ensure solvent levels are within safe limits. Lastly, we measure moisture content using Karl-Fischer titration, or KF, to assess the sample's stability. Moving on to an example of an HPLC assay, a key point is that we require a standard curve with an R-squared value of at least 0.999. This indicates a very high correlation and accuracy in our measurements. Additionally, the retention time for the analyte should be consistent, within plus or minus 0.1 minutes, to confirm the method's reliability. When it comes to acceptance criteria, the API assay should fall within 98 to 102%, ensuring the potency is within an acceptable range. And for system suitability, we look at the percent RSD, or relative standard deviation, which must be 2% or less. This helps us verify that the system is performing consistently and accurately. Overall, these tests and criteria are essential to maintaining high-quality standards and ensuring that our product is safe and effective for use. Let's talk about specifications, limits, and their justifications. Why is it important to justify these limits? Well, regulatory norms actually require us to provide a solid scientific rationale for the limits we set. This isn't just about meeting standards. It's about ensuring safety and compliance through evidence-based decisions. For example, take the impurity B limit. It's set at less or equal to 0.05%. Why that specific number? Because toxicity threshold studies have shown that at or below this level, the substance remains safe. So, the limit isn't arbitrary. It's backed by scientific data indicating a safe margin. And where do we document this justification? It should be included in our Validation Master Plan, or VMP. That way, we have a clear record showing how we determine these limits based on scientific rationale. This approach helps maintain transparency and supports regulatory inspections. Overall, justifying our specifications with scientific evidence is key to a robust and compliant validation process. Let's take a look at what a validation protocol and report should include. First, the protocol itself must clearly define the objective and scope of the validation. This helps ensure everyone understands what we're aiming to achieve and the boundaries of the study. Acceptance criteria are also critical. They set the standards that must be met for the validation to be considered successful. Next, we need to specify sampling locations and how often samples will be taken. This ensures that the data collected is representative and sufficient to support our conclusions. And of course, choosing the right statistical tools is essential for analyzing the data accurately and objectively. Moving on to the deliverables, the report should include all raw data collected during the validation process. This transparency allows for traceability and review. Any deviations from the plan must be documented, along with the CAPA, corrective and preventive actions, that address those deviations. Finally, the report should culminate in conclusions about the robustness of the process. This helps us understand if our process is reliable and consistent, which is ultimately what we're striving for in validation. All of these elements work together to ensure a thorough, credible, and actionable validation process, supporting the overall integrity of our operations. Let's look at this hypothetical example to understand how we approach challenges in production. Here, the API is called Atramax. Now, the challenge we faced was that impurity X was trending high during step 3 of the process. This kind of trend often signals an underlying issue that needs addressing. Our investigation revealed that the root cause was pH drift during the step. This is a common issue that can significantly impact impurity formation. To tackle this, we implemented a tighter pH control strategy. Ensuring more precise pH management helped us reduce the impurity levels and stabilize the process. This example highlights the importance of closely monitoring key parameters like pH and being proactive in adjustments. It's a reminder that small changes in process conditions can have a big impact on product quality, and that thorough investigation and targeted control measures are crucial in overcoming production challenges. Let's take a look at some calculations involved in quality control. Here, we have an example of a control chart. The key is to create X-bar and R charts to monitor process performance over time. When doing this, we compare the control limits, which are set at plus or minus 3 sigma, to determine if the process is stable or if there are any signals of variation. Moving on to risk tools, we see practical examples like the FMEA worksheet. This helps us identify potential failure modes and their effects so we can proactively address them. Another valuable tool is the Ishikawa diagram, or fishbone diagram, which helps us visualize root causes of problems by categorizing potential factors. Finally, we have some example statistical results. These results give us insight into process performance and help us make informed decisions. Overall, these tools and calculations are essential for maintaining quality and driving continuous improvement in our processes. Let's wrap up with some key takeaways. First, it's crucial to remember that API process validation isn't just a checkbox. It requires a solid scientific justification, thorough documentation, and statistical rigor to ensure robustness. When we think about the process flow, dispensing, IPC, sampling, and final analysis, each step must be carefully controlled and justified. Skipping or rushing through any of these can compromise the integrity of the process. And finally, adopting a risk-based, data-driven approach is essential. Making decisions grounded in data not only strengthens our validation, but is also critical for regulatory success. By focusing on these principles, we can ensure our processes are reliable, compliant, and ultimately deliver quality products. For further support, please feel free to contact us. We're here to help with any questions or additional information you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is what makes these discussions meaningful. Remember, we're always available to assist you as you apply what you've learned. Thank you once again, and have a great day.